All right, so we are starting with chapter 12 after the break. Things are a little different since I'm not there. So instead of giving you the whole packet, what you should have gotten today is the notes for today. You should also have the calendar and the review. The calendar is tentative. It can, of course, change. You guys might end up moving a little bit slower without me. That's okay. I do apologize for the lighting in this room. It's not the greatest. I will see if I can work on that, but I make no promises. Probably not gonna do a whole lot to it. Okay, so you should have your calendar and review, so you can get started on your review whenever you decide to do that. You could wait until the end of the chapter, or you could start it sooner. Um, Starting it sooner gives you more time to send me questions, since it may take me a while to get back to you. So just let me know if you have any questions as we're going through. We are going to go ahead and get started. So you don't have your homework. You can't fill it out as you're going through. Get over it. You're fine. It will actually probably help most of you. So let's get started with today's lesson. So we're doing 12.1, chapter 12. We are looking at DNA. So we're going to be talking about DNA and its structure and the process of replicating DNA. So we've already talked about the fact that DNA gets replicated before mitosis and before meiosis. So we're going to talk about how that happens later in this chapter. Today, we're just looking at how did we learn about DNA? How did we determine what DNA is and what it does? And I mean that as a very, very royal we, because it was not any of us that did it. It was a handful of experiments that were done by people probably in the I don't have your book in front of me, so I don't know the time frame for that. But I would guess in like the 40s and 50s. I was wrong. The first one was done, the first one we're talking about anyway, was done in 1928. So on your notes, what you're going to need to do is use the graphic here to fill in. I will post these online as well. I'll post the video, but I'll also post just the slides on Google Classroom, so you guys can go and access that, and you can look at the slides. If I go too fast, you can go and look at the slides, and you can fill in what you need. Okay. So in Griffith's experiment in 1928, he was looking at bacteria, and this is a strain of, I believe it's pneumonia, that there's two strains of this type of pneumonia, and sometimes one strain will kill you and the other strain does not. So he had this hypothesis that he could take dead bacterial cells and mix them with living cells, and they would kill, or they would transform the living bacterial cells to make them not the good kind of this pneumonia. Okay, so we have two strains, right? The S strain, which is considered virulent, I can't say that word very well. But that means it kills you, it's fatal. Okay. And then the R strain, non-virulent, non-fatal, not going to kill you. You might be sick, but you won't die. Okay. So he took the S strain, the deadly kind, injected it into mice, they died. Okay. So under results for injecting the living S strain, the mice died. Okay. When he injected them with the living R strain, which is the non-fatal kind, the mice were healthy. They lived. Okay. So it's in particular, this bacterial cells either infected their hearts or did not infect their hearts. So the ones that infected their hearts, the S strain, that's what killed them, is that their hearts were not healthy anymore. So he took the S strain and heated it up. And one way to kill bacteria is to heat it up. That's why you run a fever when you get an infection. Okay. That's also why washing your hands in hot water is theoretically better. In the summer, most viruses and bacterial infections are not as bad because it's hot, and if we're outside and it's hot, they get killed. by the heat and the UV rays. Anyway, heat killed this virulent S strain. And he knew that it was heat killed, knew that the bacteria was dead, because when he injected that heat killed bacteria into the mice, they were fine. They survived, they lived, everybody was happy and healthy. Okay, so that was the first three kind of experimental groups. Okay, so these were really all control groups. So he knew that the S strain was virulent and the R strain was non-virulent. And then theoretically, by killing the S strain, it should become ineffective. It shouldn't do anything. So then we get to the actual experiment part of his test. He took living R strain, remember, that's the kind that doesn't kill you, and mixed it with the heat-killed, dead, or supposed to be dead, S strain bacteria. Mixed them together, injected them into the mice. Shouldn't die, right? Because the R strain doesn't kill you, and the heat-killed S strain doesn't kill you, but the mice all died. Okay, and they found the living S strain was found in their heart cells. So what happened, right? How did that happen? And the answer is that there is some chemical substance in the S strain that didn't get killed by the heat, and instead it got picked up by the living cells. And so we call that transformation. Okay. So transformation means that we can take genetic material from one organism, in this case bacterial cells, and we can pass that genetic information to another cell. Most often this happens in bacteria. People don't do this so much, not in this way, okay? But what happens is that we have these fragments of DNA that get picked up by a cell, and then they become part of that cell, and eventually they can end up in the chromosome of that cell. Okay, so bacteria have these weird circular chromosomes that they're called plasmids. So what happened here is that this recipient cell took up one fragment of this DNA, and inserted a portion of that into its own chromosome. So we have this cell that is now carrying genes that it didn't carry before. And that's transformation. So a while later, another guy named Avery, this is his last name, I don't remember his first name, Oswald, that's what it was. Oswald Avery performed another experiment to kind of further test this idea of transformation. What he was trying to do is determine which molecule was responsible or was the most important for transformation. So you can see over here what he did. He took the heat-killed S strain, and then he mixed it with chemicals that would systematically destroy different types of molecules. So in one set, he destroyed proteins. And in another set, he destroyed lipids. And in another set, carbs. And then the RNA. And then the DNA. So in each of those in each of those tests, so for instance, this one here, he destroys the RNA. Mixes the heat-killed S strain without its RNA, mixes that with the R strain, and still manages to produce bacteria that can kill. Okay. Same thing if he destroys proteins or lipids or carbs. The only group that was different was when he destroyed DNA. If he destroyed the DNA in the heat-killed S strain and then mixed that with the R strain, and then tried to grow it, only the R strain bacteria would grow. Okay, only the, or if you had injected them into mice, only the mice would have all lived. Okay, so when you destroy the DNA, you no longer have that transformation occur. If you destroy anything else, the transformation does still occur. Therefore, he determined that it is the DNA that stores and transmits the genetic information. Okay? Because anything else didn't change the results that Griffith got. Only when we destroy the DNA do we change the results. Therefore, DNA is responsible for transformation. Avery's experiments were confirmed by another group named, it was the next experiment is involving two guys, Hershey and Chase, and they were studying bacteriophages. A bacteriophage or a phage, excuse me, stop, sorry. A bacteriophage is a virus that infects a bacteria. Okay, so a phage is another name for virus. And these guys are specifically affecting bacteria because, yes, even bacteria can get viral infections. It's true. So, viruses are very simple things. They're not even technically alive because they can't reproduce on their own. And they just, in general, don't meet the requirements for being considered a living thing. They are really just DNA, and then they're surrounded by a protein coat. Some of them really do have this kind of like robot-y structure to them. And then I'm sure you've seen the pictures of COVID where they're like round and they have like the, I don't know, to me, it looks like the like suction cups on the outside. That's what I think of. But phages, viruses come in lots of different shapes and sizes, but they're all essentially DNA on the inside and protein on the outside, and that's it. They don't have carbs, they don't have lipids, they're just DNA and protein. Okay, they can't reproduce on their own. They don't really use up energy. They would use some, but they don't like eat stuff. Okay, they just kind of exist. So the Hershey-Chase experiment was determining when a bacteriophage is infecting a bacterial cell, is it the DNA or is it the protein coat that gets injected into the bacteria? So they took advantage of the fact that protein and DNA contain different substances. They contain different elements, is the word I was looking for. DNA contains phosphorus and protein contains sulfur. Okay, so those are unique to those, between the two of them, those are unique to them. So DNA has phosphorus and protein does not have phosphorus, but protein has sulfur and DNA does not have sulfur. So they could radioactively label either the phosphorus or the sulfur and see which one ends up on the inside. Okay, so in their first experiment, or the first one we're going to talk about, I don't know which way they did it. They took and labeled radioactive sulfur. Okay, so basically they took the viruses and just kind of grew them on a medium that would have sulfur. So they would end up incorporating sulfur into, it had radioactive sulfur. So they would end up incorporating this radioactive sulfur into their protein coat. Okay, and then they mixed this now radioactively labeled phages with bacterial cells, let them all mix together, let the phages inject and infect the bacterial cells, and then they separated them. They removed the phages and kept the bacteria and measured for radioactivity. And when they were measuring with the sulfur, the radioactive sulfur, there was no radioactivity inside the cell. But then they did it again. They did the experiment again. And this time they labeled the DNA. So they were using radioactive phosphorus. And they again labeled the protein, sorry, labeled the phages, labeled the DNA in the phages, mixed the phages with the bacteria so that they could inject and infect the bacterial cells. And the way that viruses work is they inject their DNA, we now know this, they inject their DNA into living cells and then let those cells do the reproducing. So they kind of take over your cells, which is why they are bad. Because then your cells stop doing what they're supposed to and they start just making more viruses. And then eventually your cells break open and release all of these new viruses out into your body. And then you get to spread that to other people. Yay. So these phages were radioactive labeled. So they were injecting their DNA, which had been labeled, into the cells. And then again, Hershey and Chase separated the two groups. So they managed to remove the viruses. They were just looking at the the bacteria. And they found the DNA inside of the bacterial cells. Okay, so they showed radioactivity. Okay, so that's how we know that it's the DNA that gets injected. Okay, so this is a way of confirming Avery's results that DNA is responsible for transformation, because the viruses that are changing these cells, right, they're causing these cells to no longer do what they're supposed to be doing. They're instead becoming virus factories. And that is from the DNA in the virus. Okay. So those are the 3 experiments. Your task for tonight is to do the homework. Okay. It's going to ask about all 3 of these experiments, what they predicted, how they did their experiment, and then what they concluded. Okay. So let me know if you have questions. But that's the end of lesson 1.